Okay, so the second half of the citric acid cycle is going to be kind of doing just refreshing the cycle, finishing up the cycle in order to allow us to start the cycle again. So it's a little weird since it's a circle, we're going up. So reaction 5 is on the bottom. So we're going to start in the bottom here. We made our succinyl-CoA, and we're going to do a hydrolysis reaction. Okay, so a hydrolysis reaction, meaning, remember, hydrolysis means we chop it apart with water. So what gets hydrolyzed is this bond, this carbon here is what's hydrolyzed. So you'll notice it's this particular bond that we highlighted. We just made it with adding a CoA in, but now we're going to cleave it and add an oxygen there. So in this process, succinyl-CoA undergoes hydrolysis, adding a phosphate to GDP to form GTP. So this is kind of different, but it's pretty much the same thing as ADP and ATP, but it's the glutamate version, right? So instead of adenine, it's just a different base there in this particular process. So we're going to take GDP. We're going to take GDP and a phosphate, hydrolyze that bond there, remove acetyl-CoA, add in an oxygen. It makes GTP, and then that will just transfer a phosphate group to ADP to make ATP. So overall, that makes another molecule of ATP. This overall forms what's called succinate. This molecule here, you'll notice there's a star next to it. That's because this molecule is achiral. It's going to be the same on either end. Our next reaction, reaction number 6, is a dehydrogenation. So we're going to remove hydrogens. So you see we have, technically there's 2 on either of these, but let's look at these hydrogens here. These blue hydrogens that I just highlighted. So succinate is oxidized to fumarate, which is this molecule up top, through using FAD. So FAD turns into FADH2. So that's where these 2 hydrogens go, and we form a double bond. In that process of regenerating FAD from FADH2, again, we form 2 ATP molecules. So in step 5, we made 1 ATP. In step 6, we made 2 more ATP. And then reactions 7 and 8. So again, remember, we're starting on the bottom. So we have this molecule here with the double bond. So reaction 7 is going to be a hydration reaction of fumarate. So remember, fumarate is this molecule, to form malate. So we're going to take this double bond, and we're going to add water. Maybe we'll do a different color. Going to add water. So one of these hydrogens, one of them, not both, but one of them, and the OH across that double bond. We've done hydration reactions before, right? We learned that in chapter 12, maybe. I don't remember exactly which chapter, but we learned it in chapter 12, right? So we add water across the double bond. And then step number 8, our last step here, is a dehydrogenation reaction. Malate undergoes dehydrogenation to form oxaloacetate again. And remember, oxaloacetate is what then undergoes step 1 in the citric acid cycle. So these last steps, I guess we should talk about this first. So now we're going to reform oxaloacetate. So this hydrogen and this hydrogen are going to be removed in the dehydrogenation reaction, And that's, oh, I shouldn't have picked orange. Let's do a different color. How about purple? This hydrogen and this hydrogen are what gets transferred to the NADH and the NADH plus. And then you overall form that ketone here, dehydrogenation reaction or reduction reaction. In this process, again, we are forming even more ATP. So 3 more ATP out of the process to go from, or step number 8, go from malate to oxaloacetate. So part 1 of the citric acid cycle was getting rid of carbons through CO2. Part 2 is isomerizing and getting back, producing ATP to the oxaloacetate, which then can go back into part 1 in the citric acid cycle. So let's go back and look overall chemical reaction of the citric acid cycle. We have acetyl-CoA plus 3 NAD plus FAD plus GDP plus PI forms 2 carbon dioxides. So this acetyl-CoA has 2 carbons in it, right? So we form 2 carbon dioxides, 3 NADH coenzymes, reduced coenzymes, FADH2, and GDP, which transfers to ATP. So for each glucose, so for 1 molecule of glucose, this process happens twice, right? For 1 glucose. So for each glucose, we get 6 NADH plus H plus. We get 2 FADH2s, and we get 2 ATPs. So overall, that's the process that we have so far. And then remember, as we talked about those steps, the NADH and FADH2s are going to be also converted to ATP. They produce energy as well. So learning check, how many of each of these are produced in 1 turn of the citric acid cycle? So only 1, not for both. So let's look. So we, again, can just check up here, but let's go through it. We form 2 carbon dioxides. We form 3 NADHs. Remember, and that has H pluses associated as well. 1 FADH2, 1 GTP that forms ATP, just transfers that over, and then 1 CoA enzyme. Okay. So let's just do one quick review of the stages of metabolism. So what we've talked about in terms of sugars is this, what's circled in the middle. We did digestion, which takes polysaccharides down to fructose, or really down to glucose, and then glycolysis forms some ATP. Not very much, but glycolysis forms some ATP. That process forms pyruvate. Pyruvate then goes into, forms acetyl-CoA, and acetyl-CoA enters the citric acid cycle, which makes a lot of ATP and CO2, which then, which forms also NADH and FADH2 through electron transport and oxidative phosphorylation, which is the next step of, or next section of the chapter. We form even more ATP. So that's what we're going to be talking about next coming up. So this is just kind of an overview of how all of this that we're talking about fits together.